Today I am going to talk about phagocytes. As you know, the general activities of phagocyte cells are to survey the tissue compartments and discover microbes, particles such as dust, carbon particles, antigen-antibody complexes, and injured or dead cells. To ingest and eliminate these materials, finally to extract immunogenic information, antigens, from foreign matter. As you know, we have three kinds of phagocyte cells, including neutrophil, monocyte, and macrophages. Neutrophils versus eosinophil. As you know, a common sign of bacterial infection is a high neutrophil level in blood, which is called neutrophilia. Neutrophils are a primary component of pus. In contrast, eosinophil plays a minor phagocytic role in compared with the neutrophil, but it is attracted to the site of parasite infections and antigen-antibody reactions. Okay. Macrophages, if you remember, as I said before, migrated monocytes are transformed by a various inflammatory mediator to macrophages. Monocyte in the bloodstream, macrophages in the tissue. The developmental stages of monocyte and macrophages, as you know, this process is marked by an increase in size and by enhanced developmental of lysozyme and other organelles. If I need to describe it, a stem cell in the bone marrow developed to a pro-monocyte, and these cells go to the bloodstream, which is called, according to these developmental stages, called monocyte. When these cells migrated or transformed by various inflammatory mediator to tissue, which is called macrophages. Macrophages are king of phagocyte cells. As you know, macrophages, we can classify them into fixed, adhere to tissue, or wandering, moved around. هذا التقسيم يعتمد على بقائهم بسبيسيفيك أورجان، أو ممكن يتنقلون، يعتمدون على الستيج أو يعتمدون على الاميون ستيمول اللي يستقبلوه، ولذلك يضطرون أنه يتحركون المكان. The specialized macrophage, we call it a histiocyte. These cells migrate to the certain tissue and remain there during their lifespan. The name of macrophages are changeable according to the site or according to the tissue. As an example, in lung we call it alveolar macrophages, Kupffer cell in the liver, Langerhans in the skin, macrophages in other tissues like spleen, lymph node, bone marrow, kidney, and brain. Other macrophages do not reside preeminently in a particular tissue. Not only are the macrophages تشتغل كسcavenger, but they also process foreign substance and to prepare them for reaction with B or T lymphocytes. That's an example of liver tissue and copper cells of the macrophages. What's the mechanism of phagocytic discovery, engulfment, and killing? We call it phagocytosis. As you know, phagocytosis means engulfment of particles by cells, and then these particles by endocytosis. The phagocytes actually endocyte both particles and liquid substance, and this process has events including chemotaxis, ingestion, phagolysosome formation, destruction, and excretion. I think you know all of these steps before I've described them. We have a question: why phagocytosis considered not just a physical process of engulfment? Because phagocytes are also actively attacked and eliminate foreign cells with a wide array of antimicrobial substances. I'll describe these antimicrobial substances later. The first event or the first step of phagocytosis chemotaxis or ingestion. إذا ماكو signal من the site of infection ما ممكن يصير الفاجوسايتوسيس. فالفاجوسايت cells migrate into a region of inflammation attracted by أكيد أكو receptor stimulant to protect from the parasite or host tissue at the site of injury. Phagocytosis عرفناها ممكن يصير لها enhancement عن طريق الأوبسونايزيشن. الأوبسونايزيشن هو اللي شنو اللي راح يتدخل؟ هي ال process that coat the surface of microorganisms with antibodies or complement, thereby facilitating recognition and engulfment. وبالتالي عبرنا عنها عن الانهانسمنت اللي يسوي للفاجوسايتوسيس سميناه بالأوبسونايزيشن. When the phagocyte has made contact with its prey, it extended pseudopoda or pseudopodas that includes the cell or particles in a pocket and internalize them in a vacuole which is called phagosome. إذن الفاجوسوم هو شنو؟ بعد ما صارت عملية الفاجوسايتوسيس جتني الأقدام الكاذبة، دخلت البارتيكلز أو الليكويد بداخلها، صارت عملية الفاجوسوم. بعدها راح تبدي عملية الفاجولايسوزوم فورميشن اند كيلينج. اللايسوزايمز اللي هو راح يهاجر للفاجوسوم ويصير فيوجن وياهم، يسوي لي تركيبة اللي يسمونها فاجولايسوزوم اللي هي عبارة عن الفاجوسوم بلس لايزوزايم. Other granules containing antimicrobial chemicals اللي ممكن تتحرر للفاجولايسوزوم تكون لي a potent designed to poison and then analyze the ingested materials. يعني هاي الماتيريالز الانتيميكروبيال كيميكال إضافة لللايزوزايم بداخل الفاجولايسوزوم راح تعتبر مواد قوية حتى تحلل لي الميكروبز أو البارتيكلز. أوكي، هذه العملية ممكن تستمر لمدة 30 للسكافنجرز. هنا إذا تذكرون قلت أكو chemical materials لها دور قوي بالكيلينج اللي هي مسؤولة عن destruction and elimination. We have several substances يعملون مع بعض يعتبرونهم a great killing power like myeloperoxidase هو enzyme found in the granulocyte يعتبر halogen ion, forms halogen ions that represent strong oxidizing agents. hydrogen peroxidase H2O2, superoxide anion, activated oxygen, the hydroxyl free radical liberated of lactic acid, lysozyme, nitric oxide اللي هو powerful mediator, cationic protein, a number of phagolytic hydrolytic enzyme. All of these material represent a great killing power. Substances help to kill the microbes, يساعدون بالdestruction والelimination. If I need to describe the phagocytes by another way, chemotaxis, and contact and ingestion, formation of phagolysozyme granules with phagosome, killing, عمليات lysing, releasing للدبريز as a final state. Okay, we have another materials like interferon, antiviral cytokines, and immune stimulant. Interferon, a small protein produced naturally by a certain white blood cells and tissue cells that is used in therapy against certain viral infections and cancer. Okay, I think all of you know about interferon, the role of interferon as a therapy against viral infections or cancer. We have three kinds of interferon: interferon alpha, beta, and gamma. The alpha one, a product of lymphocyte and macrophages. Beta interferon, a product of fibroblast and epithelial cell, while the gamma one, a product of T cell. All are produced in response to the viruses, RNA, immune product, various antigens. In addition of antiviral activity, all three interferon can inhibit the expression of cancer genes and have a tumor suppressor effect. We have another role of interferon. Interferon act as important immune regulator cytokines that activate the development of white blood cell. Alpha interferon, produced by T cell, activates a subset of cell which is called the natural killer cells. Beta interferon plays a role in maturation of beta and B and T lymphocyte and in inflammation. Gamma interferon inhibits cancer cells, stimulating B lymphocyte, activates macrophages, and enhances the effectiveness of phagocytosis. Okay, following للphagocytosis، هذا الموضوع يعني راح أحكي أكثر مما أنه اعتمادا على الرسم الموجود. فالfollowing للphagocytosis مثل ما نعرف الباثوجين are subjected to the variety of killing mechanisms. يعني مثلا بعد ما يصير عملية اللايسوزوم فيوجن ويا الفيجوسوم راح يصير عندنا change بالpH, rise in the pH of the phagolysosome، وبعدها راح شنو ينزل الpH خلال 15 إلى من خلال 10 إلى 15 دقيقة، وبالتالي هاي العملية ممكن تأثر بعملية الكيلينج. اوكي، اكو اشياء ثانية ممكن؟ نعم اكو اشياء ثانية. production of reactive oxygen intermediated in activated macrophages which occurs immediately after internalization. هذا ايضا ممكن يأثر. ايضا الديفنسينز ممكن يأثر. زين، راح هنا نشوف هذه بحالة وجود oxygen dependent وبحالة oxygen independent. نريد نشوف شنو الفرق major differences between like resting and activated macrophages, is the ability to generate H2O2 and other metabolites generated by respiratory burst. In the presence of myeloperoxidase, higher levels of reactive oxygen metabolites will be generated. Macrophage can be activated by interferon gamma to express high levels of inducible nitric oxide INOS synthetase, which catalyze the production of nitric oxide from arginine, release of NO by macrophage, contribute to killing intracellular pathogen either directly or by production